Thank you, Dr. Rios, for introducing me. I'm very glad to be here. I was one of the organizers of the class and of the whole week, and I'm hoping that you've learned a lot and you will continue to learn a lot. We started out with some very basic lectures. I wanted you to understand genetic models and how genetic models like the Drosophila and the mouse have contributed to our understanding of cancer. And this afternoon, we're kind of in a transition phase. I invited Louise to give a talk, Dr. Strong, because she really understands the families that inherit mutations in these tumor suppressor genes. And because a lot of the work that I'm going to discuss with you today really impinges on the interactions with Dr. Strong and understanding what happens in humans, and so that I can mimic those same changes in an animal model. So my lecture is a little bit more, I guess, translational, because it's going to take some of the basic studies that you've learned about and some of the clinical aspects. And so we're going to talk about mouse models today, and we're going to talk about p53. So you've already heard about p53, but you're going to hear about it a little bit more. So this is really a very simplified slide of the p53 pathway, okay? And just bear with me a little bit. It'll be a little redundant, but I think it's important that you take home a couple of lessons from this slide. Number one, of course, p53 is in the middle. p53 is a DNA damage response protein, okay? So you, Dr. Wood this morning talked about DNA damage repair pathways. A lot of these pathways, a lot of the chemicals in smoke, UV, ionizing radiation, what those chemicals do is when they damage the DNA, they allow the p53 protein to become active, okay? So p53, David Lane called it the guardian of the genome a very long time ago, and I really like that title for p53 because that's what it does. Any type of DNA damage activates the p53 tumor suppressor, okay? p53 is a transcription factor, okay? And so that's why it's so important. It can activate, I think there's like 200 genes that p53 can activate. And in so doing, it can affect how the cell responds to this DNA damage. Okay, I think this is a pointer, maybe not. Anyway, p53 is central. And when p53 is activated, there are several responses that I'm showing on this slide, okay? One of the responses is apoptosis, okay, cell death. It makes sense that p53, there, the first one?
Yes.
I see. Okay, thank you. It makes sense that if a cell has a lot of damage and it really is not important for the organism, that it just die and not contribute to that damage to another cell. So apoptosis is a critical p53 response. Another response, which we'll discuss in a lot of detail today, is what we call the cell cycle arrest and senescence response, okay? So p53 doesn't have to kill a cell. It can stop the cell from ever replicating again. Okay, p21 is a critical target here, and it initiates a cell cycle arrest program. And p21 is also known as the senescence factor, and senescence is sort of called aging in some cases. That cell should never, definition of senescence is that cell should never divide again. Okay. There's some very recent data that I won't discuss today, but p53 also regulates some genes that are involved in how metabolism occurs in cells, and we now know that this is also an important tumor suppressive mechanism for p53. Okay, and then the last thing that p53 can do is actually allow the cell to live. Okay, and it can do this because it can down-modulate its own levels through MDM2, and Louise has already mentioned some of her studies on MDM2. Okay, so what happens is when p53 is no longer necessary, okay, when it's either allowed, given the cell time to repair and the cell's ready to proceed through the cell cycle, p53 activates this gene called MDM2, and the MDM protein, what it does is it binds p53 and targets p53 for degradation. Okay, so when p53 is initiating activation of p21, you get cell cycle arrest, senescence. When p53 is activating PUMA, NOXA, PERP, BACS, it's inducing apoptosis. But then when p53 induces MDM2, it's allowing survival because it's down modulating its own levels. Okay, feel free to ask a question at any point during the lecture. Okay, just raise your hand real high in case I don't see you. So p53 has all these different options. And believe it or not, even though this field has been studied for about 25 years now, we still don't understand how p53 can decide to kill a cell, to stop the cell from growing, or to allow the cell to, I mean, we still don't understand this. And so every cell type seems to be different. Whether it's a replicating cell or non-replicating cell seems to be affecting the response. But anyway, we still don't understand how p53 can do all this. So even though we know a lot about p53, there's still a lot to learn. Okay, so Dr. Strong already mentioned p53 germline mutations. And the first part of my talk is really going to revolve around a mouse model that mimics the human Li-Fraumeni disease that she described with you earlier. This is just a line drawing of p53, and these are the kinds of mutations that occur that are missense mutations in p53. And it's about 80% of the mutations that occur in Li-Fraumeni syndrome are missense mutations. They're single amino acid substitutions, okay? And these occur mostly in the DNA binding domain, okay? So as I told you, p53 is a transcription factor. It has to bind DNA and activate transcription of a large number of genes. And so the mutations here disrupt or dampen the ability of p53 to bind DNA and to activate transcription. These 5 amino acids are the most prevalent mutations in p53. They are arginine amino acids, and as you know, arginine is positively charged, and it's an important amino acid that can contact the DNA. So these are all arginines that are altered that disrupt either the structure of p53 or the binding of that domain to p53 targets. Okay? And I'm going to really skip the next slide because this is just it is actually one of Dr. Strong's slides. It's different colors. I guess we all have different ways of presenting the data. This is really Dr. Strong's studies. She's examined and kept up with these families for over 30 years, and she's already talked about it, so I'm just going to go through with it. I've animated this slide, and so what you'll see is this is a slide that I drew in 2003 from Dr. Strong's data, and with time, I've actually updated it. Now it's 2012, and you can see that all these additional individuals have now gotten cancer. And so that's just a real quick summary of Dr. Strong's data, and I hope you enjoyed her lecture. Okay, so Dr. Strong and I have had a long-term collaboration, and she basically tells me what kinds of mutations are occurring in human cancers, and I look at her and I tell her, well, I can model that in a mouse. So a while ago now, we made a mouse with this mutation. It's an arginine to histidine mutation at amino acid 172, and it corresponds to one of those hotspots that I showed you in the previous slide. This p53 protein is very stable in cells, as you can see here. And the main reason for making this mutation is that these missense mutations are the most prevalent kind of mutations present in Li-Fraumeni syndrome. And there was a lot of scientific data that said that having a p53 mutation was worse than not having p53, okay? And that's a study that we can actually test in the mouse. So a p53 null mouse had previously been made, and it was a very tumor-prone model. And that's good because it said that p53 was an important tumor suppressor. It prevented tumors in this animal system. So when we made this model, we wanted to see if this model was different than the model that had no p53, and indeed it was. And the most, I think, exciting aspect of this model was that the tumors that arose in this model were very similar to the tumors that arose when there was no p53 in the cell. But in this case, we saw metastasis. Okay? So having a mutant p53 led to a metastatic phenotype in this animal model, which was not present when you had a null allele. Okay? Yes?
So in the knockout, there was no metastasis?
So the p53 plus minus mouse, there's no metastasis, right? You occasionally see some micrometastases, but it's not very rampant. And then with the mutation, I call this an H allele, Arch to His. You see metastasis, and it depends on the model that you test, but it's about 30%. Okay, so not complete, and I think we understand why it's not complete, and we'll evolve into that story in just a minute. Okay. Okay, so I just want to summarize. This was a study that was published a number of years ago, but I just want to summarize what we learned in comparing mice with a mutant p53 versus mice with no p53. And the first was that the tumors from these H mice metastasized. The second was when you have this mutant protein, and it's a question I'll develop again during my lecture, is what is this mutant protein doing that it can allow the cells to metastasize? The protein is stable, and we showed that it could bind these two other tumor suppressors. They're called p63 and p73, and it inactivates those tumor suppressors. And so by having a mutant p53, you don't just lose p53 function, you're also dampening the function of two other p53 related proteins that are also tumor suppressors. Okay? And then we showed in cells that are null for p53, so they don't have the mutant protein, if you now down-modulate with siRNA p63 and p73, you now get cells that have an enhanced transformation metastatic ability in invasion in tissue culture assays. Okay? The best, I think, in vivo model was one that another colleague, Dr. Flores, developed when she was in Boston, and that is she made the genetic combinations of a p53 heterozygous allele with different p63 and p73 alleles. Okay? So p63 null mouse is embryo lethal, so you can't do the null experiment, and a p73 null mouse is pretty sickly too. So what she did is she just crossed different single alleles of these different tumor suppressors. Okay? In her study, she found a 5% metastatic rate for the heterozygous allele. Again, this was micrometastases. When she combines it with p63 heterozygosity, it's 50%. With p73 heterozygosity, it's 45%. And just inactivation of one allele of each of these two tumor suppressors gave 30% metastatic tumors. Okay? So this is a very pretty in vivo experiment that says that all of these proteins contribute to metastasis, and that what the mutant p53 is actually doing is inhibiting. Let me rephrase that. What this experiment says is if you inhibit the function of p63 and p73, you get a metastatic phenotype. And what our studies had shown is that the mutant protein inhibits the activities of p63 and p73. Okay? Okay. So we published this study, and it was a very nice study, but whenever you generate a mouse model, you always get surprises. You always get things you don't understand. And this was one of the things, and I'm going to show you two slides, of things that we didn't understand about the model. And the first was, even though I've just told you that the mice that have a mutant p53 have a metastatic phenotype, there's no difference in the survival curve of these mice compared to a null allele. Okay. So what I'm showing here is it's a survival curve. So all the mice are alive at the beginning, and then with time they develop tumors and die. So in blue here is the heterozygous mice, and in red here is the mutant mice. No difference. Okay? So, in thinking about this, we're thinking, well, maybe the mice develop metastasis, but because the primary tumor gets very large, maybe we have to sacrifice the mouse before actually, the mice die of metastasis. We don't do surgery on these mice. So that was one idea that we were toying with. It doesn't turn out to be the correct one. So let me move on and tell you what the second surprise was. So if you go to a clinic, oftentimes they will use p53 immunohistochemistry to detect mutant p53. And the reason is the mutant p53 is very stable, okay? And so when we started doing immunohistochemistry on our tumor samples, we noticed that p53 wasn't always that stable. So here I'm just showing you a small panel of the immunohistochemistry experiments that we did. We've done, here I'm showing you homozygous mutant. Okay, so there's no wild type p53 in here. These are sarcomas and these are lymphomas. And you can see here's an example where there's absolutely no stable p53. Here it's very stable, here it's not. Okay, so what's happening? Actually, 21% of these tumors did not have a stable p53, and 30% of the heterozygous tumors did not have stable p53. Okay, so we're very confused. I mean, we think mutant p53 should be stable, and it's not. So I had a terrific postdoc in my lab who just went back one step and said, let's not look at tumors, let's look at normal tissues. Okay, so here is just some of the many tissues that she looked at in a mouse. This mouse is homozygous for this mutant allele, and there is no stable mutant p53 in these cells. Okay, so that got us thinking, okay? And what we began to think is that maybe the signals that regulate wild type p53 are the same signals that regulate mutant p53. Okay, so what are some of those signals that regulate wild type p53? Ionizing radiation, any DNA damage, UV. And so the idea was that maybe in these samples, DNA damage was stabilizing that mutant p53, and the cell really didn't know that the p53 was mutant. It just wanted some p53. Okay? So the other regulator of p53 is MDM2, and Louise mentioned this in her discussion this earlier this afternoon. And I just want to show you one really pretty genetic experiment that tells you how important MDM2 is at regulating p53. A number of years ago, Roberto Montes de Ocaluna generated an MDM2 null mouse in the lab. It's actually an embryo lethal. Okay, so you can't get an MDM2 null mouse. And this is a blastocyst just prior to implantation. It's been stained with a tunnel assay to detect apoptosis, and this blastocyst is entirely apoptotic. So it implants, causes a decidual reaction, but it never becomes an embryo. It's dead. Okay, since MDM2, as we mentioned, is an inhibitor of p53, we wondered what would happen to this phenotype if you took out p53 and you completely rescue this phenotype. Okay, so now you have a mouse that's born, has no MDM2, and has no p53. Okay, critical regulator of p53, correct? So the question that we asked then was, is MDM2 regulating our mutant p53 protein? Okay. I will add that we now know that MDM2 is an E3 ubiquitin ligase, targets p53 for degradation. Okay. Okay. So we know MDM2 regulates the wild type p53, and so the question was, does MDM2 regulate the mutant p53? Okay. We had an inkling that this was going to be real. Okay. And the reason is that when MDM2 was discovered as a p53 binding protein, that cell line actually had this mutation in it. Okay? So we knew that MDM2 could bind the mutant p53, and now the question was, could it stabilize that p53 protein? Okay, MDM2 binds the amino terminus of p53, which is a transactivation domain, and that domain isn't altered in these missense mutations. Remember, these missense mutations are in the DNA binding domain. Okay, question? Somebody up there? Okay. So what do we do? So Tamara then looked at mutant p53 stability in the absence of MDM2. Okay, so here we had that panel that I showed you earlier where we cannot detect any stable mutant p53. But when we take out MDM2, you can see that every one of these tissues now stabilizes p53. Okay? So MDM2 is regulating p53, just wild type p53 and mutant p53. And now this is a survival curve. Okay? So here in black is the survival curve for the mice that are homozygous for the missense mutation. I already showed you it was the same survival curve for a mouse that had no p53. Okay? But now look what happens when you take out MDM2. If the mouse is homozygous for that missense mutation, you have a much earlier tumor phenotype. Okay? I haven't drawn it here, but in this survival curve is also the p53 null, MDM2 null mouse, because those mice have no p53 and they're also tumor prone. Okay? So we've never been able to alter that survival curve unless we have a mutant p53 that becomes stabilized when you take out MDM2. Okay? And then the last phenotype that we examined in this animal model was that of metastasis. Okay? So here, the homozygous mutant mice have about 30% sarcomas, and none of these metastasize. Okay? If this mouse is heterozygous, it does have a metastatic phenotype, but if it's homozygous, it does not. And we always thought that this was because the mouse died too quickly and you didn't really develop metastatic disease. But here, when you take out MDM2 and you stabilize that mutant p53, you can see you have about the same number of sarcomas, but now you have metastatic disease. Okay? So it's actually what is happening in the mouse is that the tumor cells begin to develop, and if at any point in development of that tumor, the cell senses that there is DNA damage, then what you have is stabilization of that mutant p53. Okay? And then once you stabilize that mutant p53, you have these metastatic effects. You can now inhibit p63, inhibit p73, do a bunch of other things, and contribute to metastatic disease. The problem is that in a specific tumor cell, we never know when it's going to become stable or not. Okay? By taking out MDM2, we stabilized it from the very beginning, and then it could contribute more readily to the tumor phenotype. We weren't counting on the cell to decide it needed to stabilize the p53 protein. Okay? Okay, so this kind of summarizes what we've learned about p53 and some of the questions that we still are trying to understand. Okay, so MDM2 clearly regulates wild-type p53, and it regulates mutant p53. And we now know that both of these proteins are stabilized in the absence of MDM2. Okay? And if p53 is wild-type, the embryo is dead, and you have to somehow figure out, you have to take out p53 for it to survive. But if it's mutant, the mutant doesn't have any activities, and so that embryo survives and develops a metastatic phenotype. Now there's some differences. And one of the things we'll discuss in a minute, well, let me just finish this slide first. Okay, so I won't discuss it today, but there's another protein that's called MDM4, and it's also called MDMX. And it's very similar to MDM2 in the sense that it also inhibits p53 activity. Okay? An MDM4 null mouse is embryo lethal, just like an MDM2 null mouse. But if you take out p53, you again rescue that phenotype. So both of these proteins are critical regulators of p53. In other models that we've generated, we also know that the interaction between these two proteins is important as a inhibitor of p53. So what we're thinking right now is that the best E3 ubiquitin ligase for p53 is actually a complex of these two proteins. Okay? Still have some work to do to evolve that idea. Okay, the other point I want to make is that if p53 is wild type, it can activate MDM2 and down-modulate its own levels. Okay? But if p53 is mutant, it's not functional as a transcription factor, and it can't activate MDM2 and down-modulate its own levels. Okay? So even though you have a cell with the wild type or mutant p53, eventually the cell with mutant p53 will have more and more of that mutant p53 because this feedback loop is missing. Okay? So MDM2 regulates p53, wild type and mutant. Everybody agree? All right. So what about other DNA damaging signals? Okay, we've learned UV, ionizing radiation, any other kind of DNA damage also stabilizes wild type p53, and the question is whether these signals are going to stabilize that mutant p53. Okay, so we're trying to really, I think the sort of the main point I want to make during this lecture is really what is mutant p53 doing in a cell to give you that enhanced tumor phenotype. So this is an experiment in which we used ionizing radiation to damage the cells. And this here is our null control. Here is what happens with this mutant. In the absence of any kind of DNA damage, you don't see the protein. You damage the cell, you see the protein. Very, very high levels. Okay? Now, I have to stop just for 1 second and tell you that each one of these lanes is a whole mouse that we irradiated and then we took out a tissue. And in this slide, it's either the thymus or the spleen, because those are the two tissues that express the highest levels of p53. And the reason we couldn't do this experiment with cells from these mice is because as soon as you take these mutant p53 cells and you put them in culture, you're stabilizing mutant p53. It's the artifact of tissue culture, the differences in oxygen between the mouse and the tissue culture dish and your incubator that activates p53 and stabilizes that mutant protein. So all of these experiments have to be done in vivo in the context of the whole mouse. And I think that's one of the reasons why we've always thought that mutant p53 was very stable. It's because you put it in tissue culture and you get a lot of that mutant protein being expressed. Okay? This is Doxorubicin. We'll be discussing Doxorubicin a little bit more, but it's a drug that's used in the clinic to treat breast cancer and other cancers. Here's our wild type p53. We know that adding Doxorubicin stabilizes wild type p53. I mean, that's the whole reason for using tamoxifen in the clinic. You stabilize the activity of that p53 and hopefully you can kill that tumor cell. Well, here's what happens with the homozygous mutant. A little bit of p53 to start with, and then you can see it's stabilized a lot in response to a drug. Okay? So stop and think about this result for just a minute. Okay? What if you're treating a patient, a Li-Fraumeni patient, that has a heterozygous p53 mutation? So one allele is wild type and one allele is mutant. Okay? Who's going to win out? Okay, is it the wild type p53 activity that's going to be sufficient to kill that tumor cell or to suppress that tumor growth? Or is it the mutant allele that's going to have a dominant effect on that wild type allele? And we know both scenarios happen, and we still don't understand why one wins out versus the other. And I think it's a very important question that we need to study in more detail. The last example I'm going to show you is reactive oxygen species. This morning we discussed oxygen as a DNA damaging agent. It's very difficult to measure reactive oxygen species, and the way we do it is with this N-acetylcysteine. So in this experiment, we're irradiating the mice Okay, and when you irradiate the mice, you activate p53, and then when you add NAC, it's a chelator of the reactive oxygen species, so you remove some of that reactive oxygen, and you can see that p53 levels go down. Okay, they don't go down all the way. Probably we didn't give the mouse enough NAC or whatever, but if you dampen reactive oxygen species, you also dampen p53. Okay, so what I think the next slide is kind of a summary slide of these data. What I want you to take home is that the cell activates p53 when it senses DNA damage, and it doesn't know whether it has a wild type or a mutant p53. Okay? So mutant p53 is just as unstable as wild type p53 under most circumstances. And in our animal model, it was tumor-specific alterations, something that happens in the tumor cell that stabilizes that mutant p53. It could be a whole myriad of signals. We have some data that I'm not going to show you today, but the RAS oncogene, for example, the MYC oncogenes, these are oncogenes that usurp the normal proliferative capacity of the cell. Both of those also stabilize wild type and mutant p53. Okay? And then once stabilized, that mutant p53 shows this gain-of-function phenotype, which is the metastatic phenotype in vivo. So mutant and wild type p53 are regulated by the same signals. Okay? Questions? Okay. Okay, so we've developed this, I think it's a beautiful animal model that says that in a specific background, the mutant p53 contributes to a metastatic phenotype and the null allele does not. Okay. The importance of the animal model compared to humans is that these are animals that are inbred to a specific background. Okay? So all of the mice that I'll describe in the next few minutes are in a C57 black 6 strain of mice. Okay? And these mice will get the osteosarcomas, they will get some angiosarcomas, they will get some lymphomas, but they will never get breast cancer. Okay? So why not? There, in this background strain, there must be some allele that is preventing the animal from getting breast tumors. Okay? So if we take this p53 allele and we put it into a different strain of mice called the BALB-C. So now you cross once and you get 50-50 C57 BALB-C. You cross again and again and again until you're in a BALB-C background. And now the most prominent tumor type that you see in that animal is breast cancer. Okay? So what this suggests is that the different strains of mice have slightly different alleles that will impact the disease that forms as a result of having a p53 mutation. We can't do that with the human population. It's a very heterogeneous population. So in the experiments that I'm going to describe to you in the next few minutes, we initiated the study looking at osteosarcomas from these p53 heterozygous mice compared to p53 plus minus mice. Okay? And the reason is that we really wanted to try to understand metastatic disease and how and what cooperates with p53 to initiate that metastatic disease. So what I'm showing you here is an expression array that is basically looking at the differences between a mouse that's heterozygous for p53 and a mouse that has a p53 missense mutation. This mouse has metastasis, this mouse does not. Okay? And you can see this is the standard kind of array data that is plotted, and there are some genes that are down modulated in the plus minus mouse that are up modulated in the mutant heterozygous mice. Okay? And there's about 200 differences between these two tumors. So we only looked at osteosarcomas because we wanted to limit the number of differences that we saw. We have a whole slew of genes. It's always the problem when you do these microarray experiments, you get too many results, and what do you look at? So the gene I'm going to tell you a little bit about is called PLA2G16. I'll tell you in a minute what it does. But there was some evidence in the literature that it could be involved in migration, metastasis, as a tumor suppressor. And so the studies that we've just submitted, so these aren't published, suggest that maybe PLA2G16 has a role in metastasis. Okay, so once we did these microarray experiments, we took a new set of heterozygous and mutant heterozygous mice and validated the data. Okay, you always have to validate your data from microarray experiments. And you can see that there is a difference. The animals that have metastatic disease and have that mutant p53 allele had a much higher expression level of this PLA2G16. Okay? Why is PLA2G16 interesting? To me, it's very interesting because it's involved in lipid metabolism. Okay? And I've already told you there's a potential role for wild-type p53 in metabolism. I think metabolism is a new and exciting area of cancer research. There's lots of evidence that how the cell manages to metabolize amino acids and other nutrients can contribute to a tumor phenotype. So this is a lipid protein. This is phosphatidylcholine. And PLA2G16 works right here, and basically it removes this fatty acid side chain. And once that happens, phospholipase D can cleave this moiety, and you get lysophosphatidylic LPA. And LPA is a signaling molecule in cells. And there's a lot of evidence that LPA can contribute to motility of cells. And for that reason, it was very interesting for us to study PLA2G16 in our system. A PLA2G16 null mouse exists, so cells can survive without PLA2G16, and the mouse is very skinny. And that's all we know about the mouse right now. But it'll be interesting to see how PLA2G16 contributes to a metastatic phenotype in vivo. Okay, so these are a series of tissue culture experiments that we've performed to try to see if it was worth making the PLA2G16 mouse. You know, you don't always commit to making a mouse. It takes a long time to make a mouse if you don't have some preconceived notion as to whether it would work or not. Okay, so here we have cell lines that express the mutant p53. And these cell lines have very high migration and invasion potentials in tissue culture. We use this little Boyden chamber that measures the ability of the cells to migrate and to invade. We used siRNA to down-modulate PLA2G16. Okay, and if you down-modulate PLA2G16, look what happens to the migration and invasion potential. It immediately drops as well. So this was a real surprise to us because I still find it hard to believe that altering a single protein has such a drastic effect on motility and invasion, but that's what the data are telling us. Okay, and in this experiment, what we did is we took cell lines that either a control shRNA or this shRNA cell lines that down-regulated PLA2G16, and we injected these cell lines into the flank of syngeneic mice to see if these would, how they would contribute to the tumor and metastatic abilities of the cell. And you can see with the control, you've got lots of tumor. With the shRNA, you have very little, if any, tumor. Here you've got one tumor that came up, and that tumor doesn't have, it escaped. It doesn't have that shRNA, so it's actually a tumor that expresses PLA2G16. Okay, so these were all really nice data that suggested that this one gene that's differentially expressed between a metastatic and a non-metastatic tumor can contribute to a tumor phenotype. In this experiment, we did the opposite, okay? Here now we have p53 null cell lines. They're osteosarcoma cells, okay? So these don't have an invasion or a migration phenotype. And now what we're doing is we're putting PLA2G16 into these cells. And what you can see is as soon as you express PLA2G16, here's an invasion assay. You can see that these cells become, they gain the ability to invade. So all those studies were done in mouse, okay? And anytime you want to publish an interesting study in the mouse, it's important to look at human samples and see if there's a similar effect in a human cell line or if this is just something that is particular to our mouse model. So here we used 2 interesting cell lines. This is a SAOS2 is a sarcoma. It's actually an osteosarcoma cell line it was developed years ago. We use it in the lab a lot. It has no p53. It has a deletion in p53. And here we have a different cell line. It's called LM7. And LM7 was developed at MD Anderson by Jeannie Kleinerman. And what her lab did is they took SAOS2 cells, they put them into mice, and they waited for tumors to develop. This cell line forms tumors very poorly, okay? And when they had a tumor that developed, they took that little tumor, grew it in culture, put it back into another mouse, and they waited till they had a very metastatic cell line, okay? So the difference, so this LM7 cell arose from this original parental sarcoma cell, but it has a metastatic ability, whereas this cell does not, okay? So here in this first panel, I show you what the PLA2G16 levels are, and you can see they're much higher in that cell line, okay? It's a human cell line. Here's the comparisons, about 2-fold higher. Here's an shRNA experiment. We can down modulate PLA2G16, and again, I think you can clearly see that these cells have migration and invasion potential in our tissue culture assays. But when you knock down PLA2G16, you lose some of that potential. Not all of it, but some of it, okay? So it's probably a contributor not just to the tumors that we observe in the mouse, but to human tumors as well. When I started talking about these experiments, I told you that in the C57 strain, the main tumor phenotype besides the lymphoma is the osteosarcomas. And now we're looking at our BALB/C breast cancer model. Same p53 mutation, but now it's in a different background. Here is just some of that data, because we never put this mutation in that BALB/C background. And you can see here, this is wild type. Here you have this missense mutation. This is the rate at which these animals get tumors. And here you have plus ionizing radiation. What we've learned a long time ago is that if you want to hurry up and get tumors more quickly, if you give the animals a small dose of ionizing radiation, that will synergize with the p53 mutation and give you worse tumors, okay? And you heard that from Dr. Strong today. Individuals with Li-Fraumeni cancer, if they get radiation treatment or whatever, they're more prone to multiple cancers more often than not. So we really just are replicating what's happening in that situation. Okay, so then we have these breast tumors, and we looked at PLA2G16 expression, and you can see here that they express very high levels of PLA2G16. And again, in these tumors, if you down modulate PLA2G16, you inhibit the migration and invasion potential, okay? So I think that this is my last slide, yeah. So what I want to suggest is that this is a protein that is differentially expressed between metastatic tumors and non-metastatic tumors. We have no idea right now how mutant p53 causes the difference, but it's only in the mutant p53 tumors. It's not limited to osteosarcomas, because these breast carcinomas also showed a difference. And through numerous experiments that I showed you briefly, if you take away PLAG16 from a mutant p53 tumor cell that has a high metastatic properties such as invasion and, migration, you decrease that, those activities. And if you add PLA2G16 back to a tumor cell that doesn't have it, you increase its ability to invade and migrate. Okay? Questions? Okay. Now, for the last few minutes, I'm going to take you again into a more sort of clinical study, because I think the mouse models are very useful for some clinical studies. And the study that I'm going to show you next is a study that was developed by Jim Jackson, a postdoc in my lab. And Jim wanted to study breast cancer, and he wanted to study the role of mutant p53 in breast cancer. Okay? So this is the paradigm. Before we started these experiments, this is how a clinician actually sees chemotherapy effect on a tumor. If p53 is wild type, DNA damage, of course, will activate p53, and then p53 can induce apoptosis or it can induce arrest or senescence. These are the two main pathways that p53 initiates to suppress tumor growth. Okay? I believe it makes sense. Mutant p53, on the other hand, can't activate the genes involved in apoptosis or arrest and senescence. And so what you should see in that case is tumor resistance. Okay. And the reason I'm telling you about this study is because this is not what we found. And it took us a very long time to figure out what was happening. Okay. So the reason that Jim started this study is because of the conflicting literature that existed in the breast cancer field. Okay? So we know p53 is mutated in breast cancers, and when it's wild type, as I've just mentioned, it is thought to contribute to drug response. Okay? But the literature conflicting. You'll read one paper and it says, yes, this is right. You'll read another paper and it says, no, it's not right. The way that scientists detect the p53 status varies between different papers. It's sometimes immunohistochemistry is used to detect the mutant p53 status, but we already know that that's not a good measure. Sequencing is probably the best way. The treatment regimes in these different individuals were different. So some got Doxorubicin, some got tamoxifen, different kinds of treatments. And then the criteria to assess response also varies widely from study to study. So anyway, the bottom line is, if you read the literature, the breast cancer literature, human breast cancer literature, p53 wild type activity sometimes is better for response and sometimes it's not. Okay, so Jim really wanted to address that in a model system. And this is the model he decided to use. Okay, this mouse expresses the MMTV Wnt oncogene. Okay, this is an oncogene that we know contributes to cancer in humans. And this mouse develops mammary tumors within a 2 to 8 month window. So this is the only alteration this mouse has. Expresses Wnt in the mammary gland and it gets breast cancer. Okay, so Jim developed this whole cohort of mice, a very large, expensive cohort of mice. And of course, some of them he doesn't treat, so he knows how the tumors grow. And then some he treats with Doxorubicin. And the first thing I'll tell you is that he had to figure out how much Doxorubicin to give the animals. And we decided that a dose that was equivalent to that present in human cancers would be a good dose. And then the other was, how often do you give it? And it's all kind of empirical. But anyway, he has a regime. And then when these animals get Doxorubicin, then he can do a whole bunch of studies. He can do short-term studies to look at p53 activity, or he can do long studies to look at clinical response. Okay? Okay, so here's just an example of some of the kinds of responses that he saw. So one tumor type is breast. These two panels in the top, no treatment. He begins the study when he can palpitate the breast cancer, because you can actually feel the breast cancer in these animals. And if there's no treatment, you can see that the tumor just continues to grow, and then we eventually have to sacrifice the mouse. Okay? Here's what happens with treatment. So all of these animals received 5, I think they're weekly doses of doxorubicin, and then he measures a tumor volume with time. Okay? So here's a tumor that was pretty large at the time of treatment. You can see a big fall in the tumor volume. It was stable for a while, and then it regressed. Okay? Here's one tumor that completely ignored doxorubicin treatment, just continued to grow. This happens about 30% with about 30% of our tumors, and we don't understand why. But we think that comparing these tumors with these tumors at the genomic level is going to be an important study, which we've not done yet, but we will be doing. Okay. But anyway, these tumors that respond are the tumors that he then followed up. Okay. So here's a tumor, has the Wnt gene, and it has a normal p53 allele. And you can see this tumor was treated 5 weeks in succession. And you can see there was a drop in tumor volume, and then you can see it regressed. So these tumors do respond, and then they regress. I think Jim animated this slide for me. Okay, so we call this stable disease, and we call this relapse. And we have samples at different time points, and so eventually, again, what we want to do is compare the changes at these different time points to see what is happening in these tumors that have relapsed. Okay. So now what I'm going to compare here is a wild-type p53 tumor and a mutant p53 tumor. And so this mutant either has the missense mutation or it's heterozygous for p53. Okay? And what we learned from this analysis is that there was a better response in p53 mutant tumors. The complete opposite of what we expected. Okay? So let's look at the graphs. Here is a response in a p53 wild-type tumor. I showed this one to you previously. Within a very short window of time, about a week, these tumors relapsed. Look at the response in the p53 mutant tumor. They have stable disease for a much longer window of time. Okay? And then if you look here, you can measure the percent tumor volume lost, and you can see that if the tumor has a mutant p53, it lost a lot more of that tumor volume. And days to relapse, 11 for wild-type p53, 24 for mutant p53. Okay, we're stunned. I mean, this is not what we would have expected. Okay, so we knew this early on, and it took us another 2 or 3 years to actually figure out why. And as you'll see, it really does make sense. But this is the study that really convinced me that Jim was onto something. So what he did in this case is he's working with the Wnt mice that are heterozygous for p53. Okay, so one wild type, one mutant allele. In about 50% of human cancers, you lose that wild type allele. And so we thought this would be a good tumor cohort to study, because some of these tumors will have wild type p53 and some will have only mutant p53. Okay, so here you can see these are all his mice. He doesn't know. When he tested these animals, he didn't know if they had a wild type p53 allele or if they had lost it. Okay, and it's hard to decipher anything from these. The tumor volumes are all over the map. So then what he did is he took these animals and he looked for LOH. Okay, and now you can see very clear differences. Can you guess? This one has a wild type p53. It is a poor responder. And this has only the mutant p53, and it was a better responder. Okay, so this is one of those situations, and we just did PCR and sequencing to look at whether those tumors retained the wild type allele, and here you can see a double peak, or whether they lost the wild type allele, and here you see a single peak. Okay, so this is one of those situations in which the wild type allele is dominant over the mutant p53 allele. And we don't understand why it works this way, but it does. Okay? Okay, so if the tumors had no p53, then they were very good responders. Okay. So then Jim had to try to figure out, okay, what was happening in these tumors. Why was the wild type p53 responding poorly? Why was mutant p53 responding well? So what Jim did was he first looked at wild type p53 tumors and compared to mutant, and he just wanted to see, is doxorubicin activating wild type p53? Okay, so he looks at p53 targets, and here he measures p21, it's a p53 target involved in senescence, cell cycle arrest, and here he looks at PUMA, which is an apoptotic target. Okay, p53 is active. Here is the treated samples, high levels of p21, high levels of PUMA. So doxorubicin treatment is activating p53 in these samples. When he looks at apoptosis, however, there's no difference between the treated and the control. Okay, so even though you're activating p21 and PUMA, apoptosis is not the readout in these tumors. And in fact, the readout is really a cell cycle arrest phenotype. Okay, so here we're measuring growth arrest by either K67 or BRDU incorporation. This is the untreated tumor, and you can see that it's it's highly proliferative. And this is the doxorubicin treated sample, and you can see it's not proliferating. Okay, so it is responding to drug treatment. And then here is the experiment that says what's really happening in these tumors that have wild type p53 and become arrested is that they're initiating the senescent program. Okay, this is a marker of senescence activity called SA beta gal. It's a common marker that the senescence field uses. Nobody understands SA beta gal. All we know is that if it's up, you have a senescent phenotype. Okay, so, pardon? Question?
SA beta gal, if it gets incorporated, it's a senescent phenotype?
SA beta gal is just, it's a chemical. Once you take the tumor out, you put it in the SA beta gal in an assay, and it measures this beta galactosidase activity. It is a slightly more acidic than normal, and that's how you can pick it out different from normal. But it's just a readout. We really don't understand what the factors are that allow the tumors to turn blue. Okay? Okay, I think my next slide shows just beautiful, you can't see it here. Take my word for it, all of these tumors, you can see the handout, it looks really good, right? Okay, so the tumors that have wild type p53 are all blue if they were treated with doxorubicin. The tumors that have one wild type allele and one mutant allele are also blue if they're treated with doxorubicin. And if they've lost that wild type allele, they're no longer blue. Okay? So what is happening in these tumors is when they see doxorubicin is they are activating p53 and they're initiating the senescent program. Okay? Okay, so senescence is a dead end. I mean, a cell that is senesced shouldn't be dividing again. But what we think is happening in this case is that only some of the cells are senescent and not all the cells. Okay, but let me give you, we know the cells are senescing. Here is the stable disease, and we're looking at senescent targets. So here's p21, which we all know is a p53 target and it's high. But these are all other genes that are also involved and upregulated during senescence, and these are also high. Okay, so we are initiating a senescent program. We're also initiating we're positive for this SASP, and these are senescence-associated secretory proteins. And this was just defined a number of years ago. But these are cytokines and the receptors of the cytokines. And I think you can see clearly this one's up, these are up. Okay. But cytokines have the opposite effect. They are promoting proliferation of cells. Okay? So what we think is happening in this case is the cells are senescing. They're all blue. And they're expressing these cytokines. And any cell that's in that tumor that hasn't senesced is now going to begin proliferating because it's got this pool of cytokines that are telling it to proliferate. So this is the model that we're working with right now. If p53 is wild type, when the tumor is treated with doxorubicin, clearly senescence. Okay, it's not apoptosis, it's senescence. Okay, the tumor shrinks, but these tumor cells begin to express this secretory-associated senescence phenotype, which is the activation of these cytokines, and these cytokines basically fuel the tumor, and within a very short time frame, the tumors grow back. Okay. And we also know that if the mouse is heterozygous for this p53 mutation and it has a single wild-type allele, you'll also see the senescence program, and you also see the activation of cytokines and the poor tumor response. Okay, quick relapse. Okay, so just the last few minutes, what is happening in tumors that lose the wild-type allele? These are our best responders. Okay. You treat with doxorubicin, you have tumor shrinkage, and you have the best response. Okay. So the first thing that Jim looked at was, of course, proliferation. Okay, and he's using probably BRDU in this experiment. Here's wild-type p53 tumors treated with doxorubicin. They stop proliferating; they're senescent. Here's the mutant p53 tumors. Here's the control; it's proliferating. Here's the doxorubicin-treated tumor. It's proliferating, okay? Which we expected, okay? Because p53 is mutant. It's not initiating a senescent or an arrest program. These cells are continuing to proliferate, okay? But they're also apoptosing at a higher frequency. Okay, so what you have here is 2 assays, cleaved caspase-3 or tunnel assays, that show that the mutant p53 tumors are dying. The cells in those tumors are dying. Here's our control, and here's the mutant p53 tumors. Okay, so why are these cells dying? They don't have a wild-type p53. They're not initiating a p53-dependent apoptotic program. They're dying in a completely p53-independent manner. Okay? So here was the answer to our question. So what's happening in these mutant p53 tumors is doxorubicin is causing double-strand breaks. Okay? DNA is damaged. There's no p53 to restore that double-strand break. These cells are not arresting in response to doxorubicin treatment. So the cells are continuing through mitosis, and when they get to mitosis, the result is a mitotic catastrophe because they've got broken chromosomes. And so actually under the microscope, what you can see, and I hope it's better in the original, is you can see this cell right here. It's in mitosis. It's trying to divide, and you can see DNA between the two nuclei that are beginning to divide, and here as well. Okay? So this is a p53 independent mechanism of killing the cell. Cell has damaged DNA, broken chromosomes, it's going into mitosis, and it's dying because it can't divide evenly the DNA between the two daughter cells. Okay? Okay. And then this last experiment actually is again looking to see if this is a possible phenotype in humans. Okay? And so what we use here is a breast tumor cell line. It's called MCF7 cells. These are a cell line that we use in the lab for a very long time. And so we took these cells. They have wild-type p53. That was important to look at wild-type p53. And when you treat these cells with doxorubicin, they turn blue. They're senescing. Okay? When you treat with an siRNA to p53, so now you're mimicking the mutant p53 situation, you're knocking down p53, you get apoptosis. And then when you look at the doxorubicin treatment and you look at the DNA, I think you can see in your handouts very clearly that you again see those same bridges between chromosomes that are trying to nuclei that are trying to divide. And if you just sort of stand back a little bit, here are the controls. You can see two perfectly beautiful nuclei separating, and you can see how aberrant and ugly these two nuclei look. So the same thing happens in a human cell line that has wild-type p53 that was treated with doxorubicin. And so we think this study then has important implications for treatment of human patients, because while both patients will respond, they're going to respond differently. And I think physicians should be aware of what pathway p53 initiates and whether it be a short-term or a long-term response. We have lots of questions. This is the first study that says mutant p53 is better than wild-type p53. We don't know if this is particular just to breast cancer. It might be particular to epithelial cancers in general. So maybe colon, maybe lung might have a similar p53 response. As I indicated at the very beginning of my talk, we still don't understand why p53 initiates a senescence program in this case, and not an apoptotic program. And I think you can also clearly see that if p53 were to initiate an apoptotic program, then that tumor, those cells would die, they would go away, and it would be a great response. So one of the things that we're also going to play with is to ask questions: what will synergize with wild-type p53 to not synergize, it's the wrong word. What will switch the pathways? What will take these cells from a senescent endpoint to an apoptotic endpoint? I think that's what we would be striving for. And so this is the last slide, -1. And what we now know is that in a tumor that expresses only this mutant p53, treatment with doxorubicin induces apoptosis because the cells go into mitosis with double-strand breaks. And that is an improved response versus the wild-type tumors. They both respond, okay? There's no question that both respond, but mutants respond better. Okay, and this is my last slide. This is just the lab a couple years ago, and Jim did the tumor study. Sue did the, Young-An Soo did the stability study. Roberto's not on here because the MDM2 mouse he made a very long time ago. Jean Lang, the p53 mutant mouse, that was a long time ago. But the other student that was in the lab is Yasmin Valentin Vega, and she's from Puerto Rico, and she was one of my star students. I didn't talk about her work today. But anyway, I want to thank you for listening, and I'll take any questions.